Hello everyone, welcoming you all to a new lecture, a new session on the med classes by the Med Mastermind. And today's topic was on microbiology, that is especially on the virology. And we will be studying a type of double-stranded DNA virus, that is adenovirus. Okay, so it is the adenovirus, a double-stranded non-enveloped virus. So starting with the topic, first of all, why are the adenoviruses are named so? Because they are isolated from the adenoids. So what do you mean by adenoids? Adenoids are nothing but they are a patch of lymphoid tissue. They are a patch of lymphoid tissue at the very back of the nasal passage. Okay, at the very back of the nasal passage, and they form a part of the immune system. Also, they form the first line of defense for the babies and young children by trapping the harmful bacteria and viruses that are swallowed in. Okay, so now what are adenoviruses? As I have already mentioned earlier, the adenoviruses are a group of medium-sized, non-enveloped, double-stranded viruses that share a common complement-fixing antigen. Complement-fixing antigen. They are non-enveloped, and as you can see here, they are having double-stranded DNA. They infect both human and animals. Okay. So, what are the general properties of the adenovirus? Moving to the size, it varies from 70 to 90 nanometer in diameter. Then, what is the shape, the symmetry, and of the capsid, and how many types of number of capsomere does it contain? The capsid is icosahedral symmetry, as in herpes virus, and it's made up of total 252 capsomeres, out of those 252, the 240 are hexon, the rest are the pentones. 240 hexon, the rest 12 are the pentones. The penton has a rod-like fiber projection. And talking about the genome, it, as I said, it's a double-stranded DNA associated with histone-like the polyp, and as it is non-enveloped, so envelope is absent. Okay, so it was the structure of your adenoviruses. Here the adenovirus, the icosahedral symmetry. Okay, is the icosahedral symmetry. Okay, it's having the DNA genome which is double stranded. The virus is non-enveloped, and it's having a hexon with a penton base and some fibers. The fibers rod-like, look, they are attached at the vertices, at each vertices, right? Here, look, at the vertices the fibers are attached, and it is the penton base on the vertices upon which the fibers are attached, and the surfaces, they are the hexon. Okay, so it is the general introduction. Here look, ये जो फाइबर्स आपका था ना, ये जो फाइबर्स जो एपेक्स से सारा आपका फाइबर्स जो निकले हुए थे, फिर आपका जो हेक्सोन था और जो पेंटोन बेस था, ये सब मिला के इस एडेनोवायरस को एक स्पेशल तरह का स्ट्रक्चर प्रोवाइड करते हैं, जिसे हम कहते हैं द स्ट्रक्चर ऑफ अ स्पेस व्हीकल. इट गिव्स द अपीयरेंस एज अ स्पेस व्हीकल. ओके, डन. Look, here it is written also. Structure of adenovirus: icosahedral appearance, hexon form the faces, pentones present at the vertices, as I have already mentioned previously. The fibers projection help in the attachment, looks like a space vehicle. Okay, looks like a space vehicle. Next, how can we cultivate, or the method of cultivation of the adenovirus? First here, what now? First of all, the laboratory animals, lab animals are not susceptible. So it is cultured in the tissue cultures of the human origin. Cultured in the tissue cultures of human origin, like your human amnion or HeLa or HEP2 cells. So what is that HeLa or HEP2 cells? We will study it. Then the cytopathic effect. The cytopathic effect of adenovirus, it includes the cell surrounding or the cell rounding and the grape-like cluster. So the cytopathic effect includes cell rounding and grape-like clustering. Inclusion bodies may be seen, may not be. So उधर हमने देखा था कोई कि क्या था वो, HeLa cells and some HEP2 cells. The HeLa is nothing but its cell line stands for Henrietta Lacks. ये हेनरिएटा का फर्स्ट दोनों लेटर और लैक्स का फर्स्ट दोनों लेटर से ओरिजिन कहाँ से हुआ है ना, देखो. द नेम इज डिराइव्ड फ्रॉम द फर्स्ट टू लेटर ऑफ द फर्स्ट एंड द लास्ट नेम ऑफ हेनरिएटा लैक्स, अ 31-year-old African American woman who was treated for cervical cancer at Johns Hopkins Hospital in 1951. The significance is that they were the first human cells to be successfully cultured, and they are known as immortal because they can divide infinitely in a lab setting. Unlike the normal human cell, they can divide infinitely. And the uses are, the HeLa cells are fundamental to microbiology, general the virus research, including the polio vaccine, HIV, COVID-19, cancer research and gene mapping. Characteristic are, they are the epithelial-like adherent cells derived from highly aggressive tumors. Okay, it's a brief introduction about the HeLa cells. And then what is the HEP2 cells? They are nothing but they are the human epithelial type 2 cells. The human epithelial type 2 cells. Originally derived from a human laryngeal carcinoma or the throat cancer in 1954, these cells were later found to be contaminated with HeLa cells, a cervical adenocarcinoma cell lining. So what is the use of this human epithelial type 2 cells in microbiology? Now they are primarily used as a substrate in indirect immunofluorescence assay to detect anti-nuclear antibodies in the patient's serum, and this test is the gold standard for identifying autoimmune disorder like SLE, systemic lupus erythematosus. Significance क्या है ना? Because the human epithelial type 2 cells are human epithelial cells, they display a wide range of nuclear and cytoplasmic antigen, allowing for the visualization of the distinct staining when viewed under a fluorescence microscope. It's of not much importance. It's just about knowing what are those cells. And then coming back to our topic, resistance of the adenovirus. Adenoviruses are more stable than other viruses and remain viable for a week at 37 degrees Celsius, readily inactivated at 50 degrees Celsius, and they resist to ether and the bile salt. Okay, they resist to ether and bile salt but gets inactivated at 50 degrees Celsius, viable for a week at 37 degrees Celsius. Moving forward, the classification of the adenovirus. So moving forward again, look, is the classification. It contains two genera, the must adenovirus and the avi adenovirus. The must adenovirus which infect the mammals, the avi adenovirus which infect the birds. They infect only the homologous species with the exception of oncogenic human adenovirus. The type 12, 18 and 31 cause sarcoma when injected into the newborn hamsters. Okay, so for birds it's avi adenovirus, For mammals, they are the must adenovirus. Then, acha. Further classification, okay. Adenoviruses are further divided into 6 groups from A to F based on the physical, chemical, and the biological properties. Antigenic structure divides the adenovirus into 49 serotypes. Antigenic property also basing upon their oncogenic characteristic, as we have seen here, based on the oncogenicity and the GC content of the DNA, and also on the heme agglutination property. Based upon the heme agglutination property, the GC content of the DNA, and the oncogenicity, they are divided into 6 subtypes from human adenovirus A to F. Okay, and its antigenic property also. About 1/3 of the 49 known human serotypes are responsible for cases of adenovirus disease. Only 1/3. Acha. So what is the antigenic structure of adenovirus? The common group specific antigen of hexon protein. They are detected by, they have a common group specific antigen on the hexon protein. They are present on the hexon protein. मतलब ये जो surface जो बनाता जो ऐसे है ना, इसी surface पे. And they are detected by immunofluorescence and ELISA test. Immunofluorescence and ELISA test से इसी antigen को हम detect कर सकते हैं. They are the type specific antigen located on the fiber proteins. Some group specific antigen जोटा हमारा अछंती hexon protein ऊपर है, and also there are some type specific antigens which are present on the fiber protein. And the common group specific antigens which are present on the hexon protein, they are detected by the immunofluorescence test and ELISA test. And the type specific antigen protein present on the fibers, the antigens, they are detected by neutralization test. And 47 types of antigenic types are known since now. Then what is the pathogenesis of adenovirus? The pathogenesis is that the adenovirus enters the host cell through the droplets, the conjunctiva or the oral route, okay, through the droplets, aerosols, conjunctiva or the oral route. Attached, then after the enter, then it gets attached to the respiratory epithelium, GI epithelium and conjunctival epithelium. So after it gets entered into the host cell, it gets attached to the respiratory, GI and conjunctival epithelium, and then it cause acute or latent infection. या तो it will cause acute infection या फिर it will cause latent infection. But the latent infection is most common in the case of adenoids and in the tonsil. Okay, make sure that you remember it. Latent infection is common in adenoids and in the tonsils. So moving further, what are the diseases caused by adenovirus? The upper respiratory tract infection, lower respiratory tract infection, ocular infection, acute hemorrhagic cystitis, then the gastroenteritis in the children, and the generalized exanthema. So slightly reading it in detail. The clinical features. Adenoviruses most commonly cause respiratory illness. However, respiratory illness most commonly, it is the most common type of clinical feature. However, depending on the infective serotype, they may also cause various other illness such as gastroenteritis, conjunctivitis, cystitis, rash illness. The symptoms of respiratory illness caused by adenovirus infection range from common cold, ये common cold से शुरू होके pneumonia तक, bronchitis तक भी जा सकता है. ठीक है? Okay. So first starting with the respiratory diseases. So what are the respiratory diseases? The most important etiological association of adenovirus is with the respiratory diseases. They are responsible for 5% of acute respiratory disease in young children and much less in the adult. Okay, 5% of the acute respiratory diseases in young children and in the adult. What happens now? 4 different kinds or the different syndromes of respiratory infection have been linked to adenoviruses. 4 different syndromes of respiratory infection. First, the acute febrile pharyngitis. It is most commonly seen in the infant and young children, including the symptoms of cough, stuffy nose, fever and sore throat. The pharyngoconjunctival fever symptoms are completely similar to those of the acute febrile pharyngitis, but the conjunctivitis is also present. Pharyngitis के साथ साथ conjunctivitis भी यहाँ पर है. It tends to occur in the outbreaks such as the children's at summer camp or the swimming pool conjunctivitis. ऐसे कंडीशन में हो सकता है. ठीक है, तो इसको याद रहा. एक्यूट फेब्राइल फेरेंजाइटिस जो हमारे इन्फेंट और यंग चिल्ड्रन में हो सकता है, और फेरेंजोकंजक्टिवाइवल फीवर in which the symptoms are completely like the acute febrile pharyngitis, that is cough, stuffy nose, fever, sore throat, but it occurs generally in the outbreaks like the children's summer camp, the swimming pool conjunctivitis. Okay, then the continuation, the fourth is the acute respiratory diseases, which is characterized by pharyngitis, fever, cough and malaise. Malaise means body pain. Before the fever there is something happening, that the body ache or body pain type, that is the malaise. It occurs in an epidemic form among the young recruits under conditions of fatigue and overcrowding. अच्छा, fatigue and overcrowding के वजह से जो youngster के अंदर epidemic condition में हो सकता है. Last is the pneumonia. Pneumonia, it's a complication of acute respiratory disease in both children and adult. ये तो दोनों में हो सकता है, in both children and the adult. Okay, so it's all about the, and then the adenovirus infections are important in military medicine. How? Look, the adenoviruses are responsible for 3 to 5% of acute respiratory infection in the children and about 2% of respiratory illness in the civilian adult. They are more apt to cause infection among military recruits and other young people who live in the institutional environment. Outbreak among the children at the boarding schools, summer camp, इन सब में होता है ना, इसीलिए 2% वहाँ चला गया. अच्छा, what is the adenovirus then pharyngitis? It is the major cause of infection associated with the non-bacterial pharyngitis and tonsillitis. The causes febrile common cold. Generally, the type 1 to 7 are the common types which are involved. Then in pneumonia, generally the type 3 and 7 are associated. This is a atypical pneumonia in the adult, but the type 7, generally the type 7, it causes serious and even fatal pneumonia in the infant. Just remember it, it may come as an MCQ. Serious and even fatal pneumonia caused by type 7 adenovirus. Okay. The acute respiratory diseases with adenovirus, as we have already studied, these are just the which types of the serotypes, or that is only informed here. The military recruits में होता है. इसको करने, इसका common agent जो associated है वो है serotype 4, 7 and serotype number 21. Then for the pharyngoconjunctival fever, the serotype 3, 7 and 14 are generally associated. It's most in the civilian population. Then the epidemic keratoconjunctivitis, it occurs in the serious epidemic. It's caused by the serotype 8 and less frequently with the serotype 19 and 37. तो more frequent is 8 नंबर वाला जो सेरोटाइप है वो करता है इसको. ये देखो केराटोकंजक्टिवाइटिस. दिस इज अ सीवियर फॉर्म ऑफ एडेनोवायरल इन्फेक्शन. इसको दिखाई देता है, ये देखो सीरियस टाइप ऑफ एडेनोवायरस इन्फेक्शन. इसको और इसमें कंपेयर करो, फिट में दिखाई देगा. ठीक है. अच्छा, next, the adenovirus in the immunocompromised patient. Generally, the patient with immunocompromised or the compromised immune system are especially susceptible to severe complication of adenovirus infection. like the acute respiratory disease, which is first recognized among the military recruit in the World War II, can be caused by adenovirus infection as condition of the crowding and the stress. And in case of the children, most children have been infected by at least one adenovirus by the time they reach the school age. At least one. किसी ना किसी एक तरह के एडेनोवायरस से तो ये इन्फेक्टेड हुए ही होंगे स्कूल एज तक पहुंचते पहुंचते. मोस्ट एडल्ट हैव एक्वायर्ड इम्यूनिटी टू मल्टीपल एडेनोवायरस टाइप ड्यू टू द इन्फेक्शन दे हैड एज अ चिल्ड्रन. एडल्ट का तो इम्यूनिटी हो गया होगा, लेकिन इनका नहीं हुआ है बच्चों का अप टू द स्कूल एज. डायग्नोसिस, एंटीजन डिटेक्शन, पॉलीमरेज चेन रिएक्शन ऐसे, वायरस आइसोलेशन एंड सेरोलॉजी कैन बी डन. एडेनोवायरस टाइपिंग भी कर सकते हैं बाय हिम एग्लूटिनेशन इनहिबिशन और बाय न्यूट्रलाइजेशन विद टाइप स्पेसिफिक एंटीसेरम. So here look the diagnosis of the adenovirus infection. The most common method used is the direct immunofluorescence, and virus demonstrated in the pharyngeal nasopharyngeal cells uses the group specific antibodies. Okay, uses group specific antibodies. Virus demonstrated in the nasopharyngeal. Direct immunofluorescence we are using. Direct immunofluorescence में क्या होता है ना, viruses demonstrated in the nasopharyngeal cells which uses the group specific antibody. Culture क्या करते हैं इनको, culture virus isolate specimen कहाँ से लेते हैं, and the virus isolated from the throat, conjunctiva, okay, urine and stool. Cell culture use के वही HeLa, then HEP2 or the KB. Characteristic साइटोपैथिक इफेक्ट आइडेंटिफिकेशन किया जाता है. देन द हेम एग्लूटिनेशन एंड सेरोलॉजी. द हेम एग्लूटिनेशन में क्या होता है ना, जनरली हेम एग्लूटिनेशन टेस्ट में हम मोंकी और रैट्स का और चूहों और बंदरों का आरबीसी लेते हैं. इट हेल्प्स इन द आइडेंटिफिकेशन ऑफ द एडेनोवायरस ग्रुप. एंड द सेरोलॉजिकल टेस्ट शोज द 4-fold rise in the antibody titer. The test use a CFT test, neutralization test, radioimmunoassay test. And for the treatment of adenovirus infection, since no specific antiviral therapy is available, supportive treatment may be done. If there will be the maintenance of hydration and the symptomatic care will be done, monitoring the respiratory complication in several infection. उसको मॉनिटर बस करना है. सो इट इज़ द ट्रीटमेंट. हाँ, इसमें होता क्या है ना, सॉरी फॉर द ग्लिच. हाँ, सॉरी. हाँ, सो इसमें होता क्या ना, द आई इन्फेक्शन में बेनिफिट फ्रॉम द टॉपिकल corticosteroids to relieve the symptoms, shorten the course of the disease. Corticosteroid इसमें यूज़ करते हैं इन्फेक्शन के केस में. Hospitalization is usually required for severe pneumonia infection and for the keratoconjunctivitis to prevent the blindness. And then prevention. Since no proper vaccine available for the general use, you can use non-attenuated vaccine for the type 4 and 7 given as enteric coated capsule in the military recruits. And non-attenuated vaccine for type 4 and 7 as it is the most fatal it can cause. It can be given as enteric coated capsule which produces subclinical infection in the intestine, and it provides the protection against the respiratory diseases in the military recruits. The same thing has been written here also for the swimming conjunctivitis. Prevention, the hand washing is the easiest way to prevent infection, and the disinfection of the environmental surface. Risk of waterborne outbreak of the conjunctivitis can be minimized by chlorination of the swimming pools. Swimming pool conjunctivitis में जो होता है, and by strict asepsis during eye examination. It's all the summary, and it is all about the completion, and it's the general. You can have a summary, the overall summary. It is important or not important, that is not So it is the completion of your adenovirus. So thank you so much. We will be meeting in another new session with a new topic, with a new chapter, and with a new zeal and enthusiasm. Thank you.